The overall goal of the following experiment is to learn about the function of aquaporins by determining the osmotic water permeability coefficient, or PF, of plant protoplasts. Cut out a piece from a leaf without abaxial epidermis and put it on an isotonic solution. Transfer the pieces to an enzymatic solution and incubate for 20 minutes in 28 degrees Celsius, then to a fresh isotonic solution. Next, collect the protoplasts containing solution with a clipped-off tip. As a second step, prepare an experimental chamber. Attach a cover slip to the bottom of the slide using silicone grease. Then, coat the chamber bottom with a positive charge-bearing glue and fill the chamber with an isotonic solution. Isolated protoplasts are then added to the chamber, and the chamber is placed delicately on the microscope stage. Subsequently, the protoplasts are exposed to a hypotonic solution while videos are recorded to capture their swelling time course. Next, the swelling time course is analyzed numerically by software, including curve-fitting procedures in order to determine the PF of each protoplast. Based on the numerical analysis, results are obtained that show differences in PF, induced, for example, by expression of different aquaporin genes.
This method can help answer key questions in the field of cellular membrane physiology, such as the regulation of the function of aquaporins.
Though this method can provide insight into osmotic water permeability of plant protoplasts that are